Mitochondria. Mitochondria live in your cells. They exist in the cytoplasm. Mitochondria have one function. They produce ATP. So mitochondria make ATP. 95% of all your body's ATP is produced in the mitochondria. That's their function. They're football-shaped organelles about the size of a bacterial cell. Okay? They have a dual membrane structure. They have an inner membrane and an outer membrane. So they have this, in other words, they have this outer membrane here which surrounds it, and they got an inner membrane which has all these invaginations in it. Okay, so an outer membrane and an inner membrane. Okay, the inner membrane is highly folded, and these are called cristae. All these little folds are called cristae. It has an electron transport system and ATP synthase. So it takes the electrons and uses the electron movement from one section to another, one molecule to another, as the energy to drive ATP synthase to form ATP. So the space between the membranes, this space right in here, between the inner membrane and the outer membrane, okay, is called the intermembrane space. Interior, let me make sure. Okay, still good. Okay, the interior is a matrix space where where your enzymes are contained. This is your interior portion. This is where all the enzymes are. Okay. You have what's called the citric acid cycle, which we're going to talk about, the beta oxidation of fatty acids, and the degradation of amino acids. The degradation of amino acids only occurs in times of starvation, like we started off saying before. So citric acid cycle, beta oxidation of fatty acids, and the degradation of amino acids. So the outer membrane, okay, has multiple pores, and that's for the passage of small molecules. The folds of the inner membrane create a large surface area. So if you have something like this, you have a surface area around it where you can have reactions occur. If you got something that looks like this, that has a lot more surface area than this one over here. Okay, all these edges now, these folds, have an area where the reaction can occur. So multiple folds creates a large surface area with many transport proteins. It allows you to transport electrons to create ATP. This is a scan electron microscope of a mitochondria, and you can see the different folds of the membrane, inner and outer membrane. This is a representation of it. You can see the matrix with all these folds inside of it. You can see the inner membrane. You can see the outer membrane. Okay. This is your citric acid cycle. It produces ATP, causes NADH. It ends up producing NADH and carbon dioxide. Okay. Also produces FADH2 and NADH as it goes through. And pyruvate, which you talked about, is our end product of glycolysis, enters into it, produces CO2 and what's called acetyl coenzyme A. The acetyl coenzyme A enters a citric acid cycle, spins through it, and produces a lot of ATP. All right, so what is the function of the mitochondria? Produce ATP. That's what it does. That's its sole function. That's what it lives for, to produce ATP. Okay. How do the mitochondria differ from other components of eukaryotic cells? Okay. Mitochondria are different than other organelles because they are surrounded by two different membranes. They have an inner membrane and an outer membrane. Okay. They have their own genetic information actually inside the mitochondria. So they make some of their own proteins and they're self-replicating. So this gives rise to the theory that mitochondria were their own organism, and there was a symbiotic relation formed during evolution between animals and mitochondria, and they incorporate themselves into it because they have their own genetic information, they make their own proteins, and they're self-replicating. So they were theorized to be alive at one point on their own, and they form this relationship with animals as we developed. Draw a diagram of a mitochondria and label the parts of the organelle. Okay, I'm not a good drawer, so let the computer draw it for me. It's not, I could have drawn that. This is a mitochondria. Okay, this is the matrix space. This is between the membranes. This is where there's increased surface area due to all these folds. So the reaction can occur in multiple surfaces. You have an inner membrane, which is this guy right here. You have an outer membrane, this guy right here. Okay, so there's your inner membrane, your outer membrane. This is your inner membrane space, is between the membranes. Okay, the increased surface area is where you have your matrix space where your reactions occur. And the cristae are these little folds that occur inside the mitochondria. That's the major function of them. That's the major parts of mitochondria. Describe the evidence that suggests that mitochondria evolved from free-living bacteria. That's what we're just talking about. Okay? Because they can replicate on their own, they're self-replicating, they make their own proteins, and they have their own genetic information. So they have everything they need to be a freestanding bacteria on their own. Now, so they infected a human host, a symbiotic relationship developed, and they got incorporated into the human body to help us produce energy. So we each use each other. Okay? So just like bacteria, mitochondria have their own DNA. They synthesize their own proteins, okay? And they grow and divide just like bacteria. They self-replicate inside our cells. The fact that mitochondria have two membranes with very different properties also suggests that we're once free-living bacteria. Perhaps the inner membrane evolved from the original bacterial membrane, and the outer membrane evolved from the membrane that captured it when it got infected into an animal species that we were developing. Okay. Now, we use acetyl coenzyme A in the citric acid cycle, and we get that from pyruvate. Remember, glycolysis, the end product of glycolysis was pyruvate. And pyruvate we convert to acetyl coenzyme A, so we can use that in the citric acid cycle, the Krebs cycle, the tetracarboxylic acid cycle, the TCA cycle. Use all kinds of cycles that spin around that help us produce ATP from acetyl coenzyme A. So under aerobic conditions now, okay, remember anaerobic was lactase and alcohol was our fermentation products. Aerobic means the presence of oxygen, okay? Pyruvate from glycolysis is completely oxidized to CO2. So you break down carbon from pyruvate all the way to CO2 now, not to alcohol, not to ethanol, not to lactate. We're going to go to CO2. So it enters the mitochondria and is converted to acetyl coenzyme A. So inside the mitochondria, pyruvate is converted to acetyl coenzyme A. Activates the acetyl group for entry into the citric acid cycle. Once it goes to the citric acid cycle, we have the Krebs cycle, tricarboxylic acid cycle, and the TCA cycle. Right. So acetyl coenzyme A, it's a very important molecule. Pyruvate enters the mitochondria. After glycolysis, we break it down to pyruvate, that goes into the mitochondria. It's converted to a 2-carbon acetyl group. Okay. To undergo more reactions after this, you have to activate it. You got to be able to do the next activation. Activation occurs when the acetyl group is bonded to the thiol group of coenzyme A, which is a high energy bond. That's the key. When we bond to high energy bonds, you can use that bond to activate that to process it more in the cycle as it goes into. So the acetyl group is bonded to the thiol group. Remember, thiol is a sulfur group. Thiols are SH, alcohols are OH. Okay, so thiols are an SH group. The SH bonds to the acetyl group of pyruvate, and we're allowed to convert that into acetyl coenzyme A. Coenzyme A is a large thiol derived from ATP and pantothenic acid, and it is a vitamin. Oops, just give one now. Okay. So this is your acetyl. This is your thiol group attachment here. This is the acid attachment of pantothenic acid, and this is a phosphorylated ADP. So here's your ADP is a diphosphate. Okay, this is adenine. Okay, this is your acid, your long-chain hydrocarbonic acid. This is amino acid. Here's your peptide bond. Okay, and this is the acetyl group that gets attached to the thiol group. This is acetyl coenzyme A. Okay, there's an overall decarboxylation and oxidation of pyruvate. So pyruvate, again, in the mitochondria, goes to acetyl coenzyme A as it enters the mitochondria. Right? You get decarboxylation, which means the loss of a carbonyl group, and it becomes CO2. Get oxidation by NAD+, which accepts the hydride ion, and it goes to NADH. The remaining acetyl group linked to coenzyme A is a high-energy thiol ester bond. It attaches to the end to that SH linkage and becomes a high-energy bond. Reactions catalyzed by 3 enzymes and 5 coenzymes bundled together as a pyruvate dehydrogenate complex. So you have these enzymes that the 3 enzymes and 5 coenzymes come together and they bond it. They form a pyruvate dehydrogenase complex. Okay. So you have pyruvate, which is your 3 carbons, and you get decarboxylation, which is going to form CO2, and it's going to bond to the thiol group of coenzyme A. Okay. So this is your coenzyme A here, acetyl coenzyme A, because here's your acetyl group. This is your coenzyme A. This is your sulfur bond. And attached to that is the acetyl group right here, these 2 carbons. Okay. And you're generating CO2. And you're taking NAD plus and converting that in NADH as the electrons are transferred. So NAD plus is being reduced to NADH. And this is being, like I said, reduction occurs without oxidation. This is being oxidized to CO2. So pyruvate is being oxidized and NAD plus, is being reduced. So you need pyruvate, coenzyme A, and you got acetyl coenzyme A. So the pyruvate dehydrogenase complex, again, contains 5 coenzymes, 4 that are vitamins, and 3 enzymes that make up this one group. You get a thiamine pyrophosphate, which is thiamine. You get FAD, which is riboflavin, the vitamin. NAD plus, which is niacin. I think it's vitamin B9. Coenzyme A, pantothenic acid, and lipoamide. Okay. So again, pyruvate is oxidized to CO2. Then you get this pyruvate dehydrogenase complex, where NAD plus gets reduced to NADH, and then coenzyme A bonds with, from here, the acetyl group from pyruvate, and you end up with acetyl coenzyme A, which is activated now because it has a high energy bond attached to it. So now it can continue on in the reduction process to reduce energy. Okay. The role of coenzyme A, acetyl coenzyme A. Okay. So acetyl coenzyme A is necessary. It's central in the cellular metabolism for the cell to function. So major function is to carry the acetyl group to the citric acid cycle. The citric acid cycle breaks down the carbon in the acetyl group to produce energy. It also functions in the biosynthetic reactions to produce cholesterol and fatty acids. It permits the interconversion of energy sources. Okay, so it converts fats, proteins, and carbohydrates to energy. That's what acetyl coenzyme allows it to do. Okay, allows you to break down, because there's 3 types of nutrition you're taking in your body: fats, proteins, and carbohydrates. And it takes all 3 of those sources through different methods now. And it's going to use acetyl coenzyme A to break those down to provide energy for your body. Okay. It produced energy by degrading glucose, so it breaks glucose down into glycolysis, pyruvate. Pyruvate goes through the citric acid cycle by becoming acetyl coenzyme A. Fatty acids, it's going to break down. Okay, and some amino acids, it's going to break down. Okay, so acetyl coenzyme A is made from polysaccharides. These are your monosaccharides. This is glycolysis, breakdown of sugars. Okay, sugar makes acetyl coenzyme A through pyruvic acid. So glycolysis breaks down glucose, glucose-6-folate, glucose-1-phosphate, phosphorylase, moves around, breaks it down, splits it into pyruvate. Pyruvate is converted to acetyl coenzyme A in the mitochondria. Lipids. Lipids are broken down into fatty acids. It breaks the lipids apart and it forms acetyl coenzyme A as it breaks lipid apart into portions. Okay, it breaks into its fatty acids, then breaks into acetyl coenzyme A. Proteins. Proteins break into amino acids, which are converted to acetyl coenzyme A. So acetyl coenzyme A breaks down every single nutritional thing that we have. Because when we eat, it's either going to be a sugar, a polysaccharide, carbohydrate, a lipid, which is a fat, or protein. Right? No other choices. And they break down every one of those to acetyl coenzyme A. Now, acetyl coenzyme A can be used in the citric acid cycle, which is a tremendous source of ATP, breaks down to carbon dioxide and water. So we're completely oxidizing a carbon compound, and we're producing ATP. We can also produce cholesterol. Cholesterol is used in forming our cell membranes. Okay? If too much of it is produced, then we have a problem though too. Cholesterol is used for forming steroids in the body and our bile salts. It produces ketone bodies, okay, which we can measure to a different test, and it produces fatty acids, which form our triglycerides and our phospholipids. Phospholipids, again, are used in forming our cell membranes. Triglycerides are a storage form of fat. We can use it to store as energy. If we have too much, triglycerides build up, or these cholesterol molecules build up, and we have atherosclerosis. We have hardening of the arteries because this plaque from these extra fat molecules build up in our body. Okay. So what vitamins are required for acetyl coenzyme A production from pyruvate? So we're taking pyruvate, okay, and we're going to acetyl coenzyme A. Pyruvate is formed from glycolysis, breaking down the carbohydrates. Okay, so pyruvate is converted to acetyl coenzyme A by pyruvate dehydrogenase complex in the mitochondria. Remember, it gives out carbon dioxide and forms that complex. Okay, then it begins and bonds to the coenzyme A and becomes acetyl coenzyme A through a high energy bond with the thiol group. So this huge enzyme complex requires maybe 4 coenzymes, okay, and different vitamins, each of which is made from a different vitamin. The 4 coenzymes are thiamine pyrophosphate, made from thiamine, FAD, which is made from riboflavin, NAD+, made from niacin, and coenzyme A. So those are all come together in that complex. Okay, this huge complex, and that forms acetyl coenzyme A. Okay, the coenzyme lipoamide is also involved in this reaction too. So what's the major role of coenzyme A in catabolic reactions? Okay, the major role of coenzyme A, okay, carries 2 carbon acetyl groups to the citric acid cycle. Remember, bonds to it and allows the citric acid cycle to spin. Their acetyl group is completely oxidized and large amounts of ATP are made. That's what coenzyme A does. It allows the citric acid cycle to circulate and it can produce a large amount of ATP from your acetyl groups where you got from glycolysis where it's broken down.
Do we need to know the intricacies of the Krebs cycle?
We're going over it right now.
Okay.
You know, the basics of it, yeah. Not
Yeah.
exact thing, but to be able to identify it.
Okay, yeah, we did this in micro. Well, I did it in micro.
Yeah, it's gonna be the same thing.
Okay.
It doesn't change.
I just wasn't sure, like, it was going to be one of those things like, hey, like the Krebs cycle produces this much ATP, and or if it's like all 27 steps.
Well, no, only we got 10 steps with it, but you'll show the ATPs that are produced.
Okay.
Okay. So aerobic respiration. Remember, aerobic means oxygen. So it's oxygen required breakdown to produce ATP, because oxygen is going to bind with hydrogen to form water, and that's going to allow the hydrogens that run out of energy to regenerate and allow them to generate a lot of ATP. That's what oxygen is required to generate a lot of ATP. So the process is also called oxidative phosphorylation. Oxidative phosphorylation is the same thing as aerobic respiration. Oxygen is required. Okay. So oxidative phosphorylation is energy from oxidative reactions. You're oxidizing the carbon structures. Is used to phosphorylate ADP to ATP. So you're adding a phosphate group to ADP, making it from adenine diphosphate to adenine triphosphate. And ATP is our energy source of the cells of the body. Okay, it's performed by enzymes in the mitochondrial matrix. So once it's in the mitochondria, you have a lot more ATP. Like when we take NADH, NAD plus NADH generates 2 ATPs normally. Inside the mitochondria, it generates 3. So things are different in mitochondria because you have the electron transport chain in there. You have 3 oxidative transfers hydrides to 3 oxidations, transfers of hydrogen to NAD plus or FAD. Both those get reduced. You get hydrogen added to them. And energy can be generated from those reduced products because they can be oxidized again. Electrons pass from NAD plus or FAD to the electron transport chain. Once it goes electron transport chain, and then oxygen uses electron transport to combine with hydrogen to form water. And that's what generates ATP, kind of indirectly. It goes down the electron transport chain. So protons are transferred to intermembrane space, leading ATP synthesis as protons, return to the mitochondrial matrix. We'll give you an exact picture of how that works. Okay, so what is meant by oxidative phosphorylation? Okay, we said it's the exact same thing as aerobic respiration. So oxidative phosphorylation is a process in which energy of electrons is harvested by the oxidation of fuel molecules, which are your carbon molecules, your pyruvate, what glucose is broken into, is used to phosphorylate ADP to ATP. That's in the presence of oxygen. Because oxidation has to occur, means you're adding oxygen. Okay. What does aerobic respiration mean? Same thing. Aerobic respiration, oxygen requiring degradation of few molecules to produce ATP. You get oxygen requiring. That's aerobic respiration. Now, the citric acid cycle, go through this again. There's several steps to it. Okay. So the citric acid cycle is the final stage of the breakdown of dietary nutrients. So everything's going to break down, whether it's a protein, fatty acid, or a carbohydrate. They're all going to break down and enter into the citric acid cycle. Then we go glycolysis to break down glucose. That forms into pyruvate. And pyruvate comes down with acetylcoenzyme A to enter the citric acid cycle. Okay. Acetylcoenzyme A and oxaloacetate feed the citric acid cycle. So acetylcoenzyme A bonds to the pyruvate, gets it started. Okay. Oxaloacetate is the first ingredient or first component in the citric acid cycle. So when the citric acid cycle spins, it starts with oxaloacetate and it has to end up with oxaloacetate being reproduced because you got to keep that cycle able to go again. You got to keep it running. So if we're starting with oxaloacetate, we have to finish with oxaloacetate so we can reuse it again and keep the cycle going. Does that make sense? Sort of. Okay.
Yeah.
Yeah, it makes sense.
Okay. So the acetyl group is oxidized to 2 molecules of carbon dioxide, completely oxidized. High energy electrons are transferred to NAD plus and FAD, where they become NADH and FADH2. Okay, and those are going to produce a certain amount of ATPs from that reduction also. So the cycle comprises 8 enzymatic steps, which are allosterically controlled, which means they're inhibited by a binding on the enzyme, not on the active site. It binds to a portion of the enzyme away from the active site and shuts it down. In other words, if the body has a lot of ATP running around, the ATP is going to allosterically bind to the enzyme and say, hey, we don't need more ATP right now. And it's going to slow the cycle down. Okay, there's no sense producing stuff we don't need. Okay, so the first reaction, okay, and this is going to start with oxaloacetate. Like we said, it starts with oxaloacetate and has to end with oxaloacetate. Okay, so a condensation reaction, remember condensation means adding water to it, adding H and OH between the acetyl group of acetylcoenzyme A and oxaloacetate. So oxaloacetate and acetylcoenzyme A. And you can add water across those peptides. And we're going to use the catalyst of citrate synthase. Okay, and you can produce citrate and you're going to regenerate coenzyme A. So they can go back again and bond to another oxaloacetate and start the process all over again. Okay. So now we've got citrate being produced. That's our first reaction. So the product is citrate. Second reaction, we start with citrate. which you just produced. A dehydration reaction means you're going to lose water between it. Okay. Followed by hydration to isocitrate. You take water off and put water back on. Okay. Hydration of cis aconitate is to isocitrate. It's catalyzed by the enzyme aconitase. The intermediate step is cis aconitate, and the final product is going to be isocitrate. So there's citrate, there's your enzyme aconitase, and you form your intermediate as cis aconitate. There's your intermediate. Okay, same enzyme is used. You're going to rehydrate it, and you're going to form isocitrate. And what it's done is switch the positions of the H and the OH. You can see where they started off over here, and the positions have been switched. Okay. So it's just an isomerism. Okay. Same chemical formula, different structural formula. It just switched to pre-action. The third reaction, we're going to start with isocitrate, which was produced in the second. Okay. So the first oxidative step of the citric acid cycle is the third reaction. Okay. Hydroxyl group of isocitrate. Okay, it's oxidized to a ketone. So here's your OH group, and it's going to become oxidized to a ketone. You've added more bonds to oxygen. Okay. Carbon dioxide is released. Okay. In decarboxylation, NAD plus is reduced to NADH. And NADH is going to form more ATP. It's catalyzed by the enzyme isocitrate dehydrogenase. And the product formed from isocitrate is now alpha ketoglutarate. So this is your alpha ketoglutarate. Okay. All it's done is form a carbonyl group, a ketone basically, where the OH group or the alkyl group used to be. And you formed alpha ketoglutarate. So the next step is going to start with alpha ketoglutarate. Okay, which it does. Alpha ketoglutarate was a product of lysine. So the next step 4 is to start with that. So now coenzyme A attaches to the alpha ketoglutarate, okay, and it reacts similar to that of pyruvate dehydrogenase complex. That's like we did before, that big complex, and then breaks down to NAD plus, and then the succinyl coenzyme A. So the enzyme involved is alpha ketoglutarate dehydrogenase, and the alpha ketoglutarate dehydrogenase forms a complex. It binds with it and forms a complex with the coenzyme A and the alpha ketoglutarate. Okay. Alpha ketoglutarate is then oxidized with NAD plus being reduced to NADH. Again, we're forming more NADH, which is going to form more ATP. Coenzyme A combines with the succinate, okay, to form succinyl coenzyme A. Bonds between succinate and coenzyme A is a high energy bond. It's this bond right here, like a phosphorus bond. It's a high energy bond. We break it to produce energy to form things in the cell. And our product here is succinyl coenzyme A. So, oops, I did go too far. Reaction 4, here we go. Reaction 5. Okay, so succinyl coenzyme A is our starting point for reaction 5. Okay. Succinyl coenzyme A is converted to succinate by the enzyme succinyl coenzyme A synthetase. Okay. High energy thioester bond is hydrolyzed, and inorganic phosphate is added to GDP, guanine diphosphate. So you have guanine diphosphate and plus your phosphate. When you break this high energy bond, the energy released from that is going to take GDP and form GTP. Now GTP is going to convert to ATP. It's going to convert ADP to ATP from that energy bond. So it's a high energy bond that's being broken, and you're forming GTP from GDP. Okay? And you're also forming succinate from succinyl coenzyme A. So you have succinate, GDP, and then you're re-releasing coenzyme A. Okay. Now the second part of step 5 is that GTP that was produced uses that high energy bond, that phosphorus bond, to bind with ADP and convert it to ATP. So one GTP goes to one ATP. Okay. The enzyme that does that is dinucleotide diphosphokinases. Again, you don't have to memorize all these enzymes, but be familiar with it. So when it's asked on the test, you can look it up. You'll know where to find the information. You know where the the citric acid cycle is. Okay, and what's involved in, and know what to look for. So understand it, but you don't have to memorize every single enzyme in here. Okay, so now you produced ATP. The sixth step, succinate. Okay, that's what we produced in the end of the fifth one. So succinate then becomes oxidized by succinate dehydrogenase, that's your enzyme, to form fumarate. And there's fumarate. Okay, and FAD is reduced to FADH2. Remember, when something's being oxidized, something has to be reduced. And when reducing FAD or reducing NAD+, we're allowing those bonds to transfer, and those can convert into ATP when those electrons are released again and go down the electron transport chain in the mitochondria. Okay. So oxidizing agent FAD, which is flavin adenine dinucleotide, is reduced in this step to FADH2. The seventh reaction in the citric acid cycle, reducing the double bond of fumarate. Okay, again, fumarate is what we're starting with. That's what we ended with in step 6. And hydration reaction, we're going to add water. Okay, reaction is catalyzed by fumarase. Remember the ASE ending is an enzyme, and it's going to produce malate. Okay, and again, we added an OH, we added water to it. So we've oxidized that. Increase our bonds to oxygen. Reaction 8, the final reaction step uses malate dehydrogenase. Okay, and there's your malate to produce, and guess what it produces? What we started with, oxaloacetate. So we regenerate what we began a cycle with, so the cycle can continue. You start with it, use it at that, and then regenerate at the end, so we can reuse it again. That keeps the citric acid cycle steady. And we produce another NADH by reduction. Okay, and NADH is going to produce ATPs. Okay, each NADH is going to produce 2 ATPs if it's outside the mitochondria. Inside the mitochondria, it's going to produce 3. Okay, so this oxidizes malate to oxaloacetate by malate dehydrogenase, and we reformed oxaloacetate, making a full circle. Ta-da! And that's it. That's what it looks like. Okay, so those are all the steps. I say you don't have to memorize all the parts to it, but if I say what enzyme catalyzes fumarate to malate, you should be able to look at the chart and realize it's fumarase. That's what I want you to do. I want you to understand the citric acid cycle. I don't want you to memorize every part of it, but I want you to understand it. So if a question's asked, you go, hey, I know where to look, and I understand what that means. That this NAD is going to NADH, that GTP goes to ATP. FADH goes to FADH2. Those are the reduced forms, and now they have energy. They can release those electrons and go back down to the original state by putting them in the electron transport chain, which we're going to talk about in just a little bit. Okay? And this all starts when glucose glycolysis. Okay, so from glycolysis, remember, when glucose is broken down, we form 2 pyruvate molecules. So we're going to get 2 spins of this thing for every glucose molecule, because we're getting 2 pyruvates. Okay? And pyruvate is going to go to acetyl coenzyme A, and that's going to start the citric acid cycle by reoxidation with oxaloacetate. Those are the points I want you to know. Control the citric acid cycle. So as with glycolysis, the citric acid cycle responds to the energy needs of the cell. If the cell needs energy, ATP is low, it's going to run. If the ATP content is high, it's going to be inhibited. Okay? You can't just spend time making one thing if we already have it. So pathways speed up when a greater demand is needed for energy. 4 enzymes, enzyme complexes, are allosterically regulated. That means they bind to a different spot on that enzyme. So there's different enzymes that control the citric acid cycle. There's 4 different enzymes in there that have allosteric inhibition. So if one of those products builds up, it's going to bind to a site away from the active site and alter the active site so it's no longer active. That's what allosteric regulation is. And that's going to slow down or stop the citric acid cycle temporarily. So several regulated studies demonstrate the importance of precise control. And you have to have precise control. You got to be able to generate energy, and you got to be able to stop doing it to other things that the cell needs. Okay? And here's the 4 allosterically regulated reactions. Conversion of pyruvate no, excuse me, to acetylcoenzyme A. It's inhibited by high concentrations of ATP, acetylcoenzyme A, and NADH. Okay, if you have a lot of acetylcoenzyme A, you don't need to convert it to acetylcoenzyme A. Okay, you have a lot of ATP, you don't need to run the Krebs cycle. A lot of NADH, you're going to generate a lot of ATP from that. You don't need to run the Krebs cycle. So it binds to the enzyme that converts pyruvate to acetylcoenzyme and inactivates the enzyme. And you got to remember, it's not binding to the active site, so it's not going to compete. It's not going to be a competition reaction. It's binding to an allosteric site away from the active enzyme site. And the second one, synthesis of citrate from oxaloacetate and acetylcoenzyme A. Negative effector is for high levels of ATP. Again, high levels of ATP, it's going to bond on it, and it's going to stop the oxaloacetate from bonding with acetylcoenzyme A. Third step, oxidation and decarboxylation of isocitrate to alpha ketoglutarate, okay? Forming a ketone from an alcohol. Okay, you're going to stop that step from occurring. It's a positive effector. ADP controls it, okay? So if energy, this is a positive effector now. The other ones are negative effectors to shut it down. This is still going to shut it down, but from a positive control. So when ADP is high, okay, it's inhibited by high levels of ATP. It's a positive effector from ADP, what controls this enzyme. So when ADP, okay, it increases the, I'm saying this. When ADP is high, it takes that control off that enzyme and lets that enzyme go through because you want to make more ATP. Okay, so high levels of ADP cause this reaction to have a positive effect. In other words, it takes the binder off the allosteric site, allows that active site to open up, and allows reaction to continue. So we can control it positively and negatively. The first we talked about were negative control. We stopped it. But if ADP builds up, if you have high concentrations of ADP, that means we've used up a lot of our ATP. So high concentration here, you want to run the Krebs cycle, and it's going to control it to increase the rate of the Krebs cycle. Where it's going to be inhibited, negative control by NADH and ATP. If those are around, then that has a negative control on the cycle and slows it down. Okay. Conversion of alpha ketoglutarate to succinylcholine is inhibited by high concentrations of ATP, succinyl coenzyme A, and NADH. Okay? All of those are already present. That step's already gone through. So if you have high amounts of the product, you don't need more of it. You have a high amount of ATP, don't need more of it. Don't need to keep running. High amount of ADH, you don't need to keep running. So every single reaction, all four, are negative controls except the oxidation and decarbonation of isocitrate and alpha glutarate. That one has a positive effector in ADP. When you think about it, if there's a lot of ADP, it means you need energy because you got to convert that to ATP. That's the one positive effector on it. And those are the four allosteric enzymes. And again, this is regulation of pyruvate dehydrogenase complex. This is a citric acid cycle, as it spins, and it just doesn't put the enzymes on there. But it's the same thing we looked at before. But pyruvate breaks down to acetylcoenzyme A, and then oxaloacetate, and starts to the citrate and starts our reaction. And this oxaloacetate is regenerated from malate as this carbic acid, as a citric acid cycle spins. Okay. Oxidative phosphorylation, transferring a high phosphate bond in the presence of oxygen. So the respiratory electron transport system. So when you're forming NADH and you're forming FADH2. Okay, basically you're transferring electrons. It's an oxidation reduction reaction. These are being reduced, so electrons are being transferred. You're going to use those electrons to generate ATP by passing them through the electron transport system. Okay, it's made up of a series of electron carriers that are embedded in the inner mitochondrial membrane. That's your electron transfer system takes place on the inner membrane of the mitochondria. Okay. So all three sites, electron transport system, okay, proton is H+, can be pumped from the matrix inside the membrane to the inner membrane space. Remember that space between the membranes? That's where you're going to pump these protons to. So when you're in the mitochondria, NADH, when it pumps its electrons, is going to provide enough energy to make 3 ATP molecules. FADH when it's inside the mitochondria, is going to pump enough electrons to make 2 ATP molecules. Now, all you got to remember is NADH, when it's outside the mitochondria, only gives you 2 ATPs. That's what's going to come when we're going to do our calculations. Okay. So ATP synthesis occurs at the ATP synthase. Okay, that's the thing that allows ATP synthesis to occur. Okay. So it's occurring in the mitochondria. So the NADH and the FADH2 that have been produced are now going to be used to transfer those trapped electrons to the electron transport chain found inside the mitochondria. And that's going to produce more ATP. Okay? So an electron transport system. So embedded within the mitochondrial inner membrane are the electron transport system. These carry electrons. They move electrons from one place to another and absorb enough when they're moving down that chain to produce enough energy to convert ADP to ATP. Okay? And these carriers are coenzymes, which you've seen, cytochromes, and they're arranged to allow them to pass electrons from one carrier to the next. So they hand it off from one carrier to the next carrier. As it goes down, that's what's called a chain. It's like a chain link as it goes around this chain. This array is called the respiratory electron transport system. Respiratory because it requires oxygen. Okay. You have a hydrogen ion gradient. So you're pumping H plus from the NADH and the FADH2. So at 3 sites, protons, which are protons just H plus, that's a proton. Okay. Can be pumped from the matrix of the mitochondria to the inner membrane space of the mitochondria. These protons contribute to high energy H plus reserves. Okay? It's pumped against a gradient, so therefore you have a potential of energy now because you're going against a gradient. Okay? Each site pumps sufficient protons to produce 1 ATP molecule. Okay? And you have 3 different sites, so it allows you to have 3 ATPs for NADH. NADH dehydrogenase passes electrons along all 3 sites, so therefore you get 3 ATPs. Okay, FADH2. Okay, oxidation passes electrons only at 2 sites, so each FADH2 produces 2 ATPs. So NADH uses all 3 sites, therefore generating 3 ATPs inside the mitochondria. FADH only uses 2 sites, therefore only generates 2 ATPs. So the final component of oxidation phosphorylation is ATP synthase. That's the final step. Okay, that is formed. It spans the inner membrane, serving as a channel for proteins. Remember, it has a channel inside the cell that allows it to go across that gradient from inside the cell to outside the cell. That's what channel is created. A portion of the complex is an enzyme that catalyzes the phosphorylation of ADP to ATP. So you put a high energy phosphate bond on ADP to form adenosine triphosphate. And this is the electron transport chain. These are the sites that it goes through. Okay. Again, you have 3 sites. Right here is it pumps H plus across. So NADH uses every one of those sites. FADH2 only uses these 2 sites. Okay. And at the end, the electrons are kicked up with oxygen to produce water. And that kicks the last energy to get high levels. And these electrons travel around through this channel, going from FO to F1, and that phosphorylates ADP to ATP. That's a real basic summary electron transport chain and the different sites involved. Okay, then just remember, this is the last site right here. This is our ATP synthase. FO, F1 complex, when it come together, it goes through the channel, and that's what gives the energy to form the ADP to ATP. Okay. So NADH carries electrons. Remember, it came from glucose when it first started on pyruvate to the first carrier electron transport system, NADH dehydrogenase. Okay. So NADH is oxidized back to NAD plus. Okay. So it takes its hydrogen, gives it an electron transport chain, and comes back to NAD plus, which can then again be used when oxidation occurs in the citric acid cycle to form more NADH. Okay, so it kicks right back into the citric acid cycle. We go NAD plus, go back to NADH again. A pair of electrons pass to the next electron carrier, Our hydrogens are pumped to the intermembrane compartment. That was the space on the side of that membrane you saw. It pumped it out of the cell into the intermembrane component. Okay. Then once it's in the intermembrane component, those electrons in the form of hydrogen are now passed through the electron transport system from one to the other and generate H+, which runs through the final channel at the end, the FO-F1 complex, converting the ATP synthase, converts ADP to ATP. Okay. So with each transfer, electrons lose some energy. So the energy to transport H+ across the inner membrane, that's the energy involved to transfer that against a gradient. That's what's giving it its potential energy to form more ATP. It's going against that gradient. So electrons reach the last carrier with too little energy to affect the transport. So you have to have an electron acceptor. Something has to accept those electrons, and that is oxygen. That's why oxygen is so important. It accepts the electrons to get that last step over the hump so it can go and create ATP. So we take oxygen plus H+, or protons, to form water. Okay. Remember the byproducts of respiration are water, okay, and CO2. Remember the CO2 breaks down during the citric acid cycle, okay, with complete oxidation of pyruvate. So now the protons, your H+, that are in the intermembrane space flow back through ATP synthase. That's that little big channel that funnel things down through the cells that converted ADP to ATP. Okay, so it's an FO channel activating F1. F1 catalyzed phosphorylation of ADP to produce ATP. All right, so this is our energy yield. So glycolysis. Glycolysis is glucose breaking down to 2 pyruvates. Okay, remember it took 2 to start it, to get it kicked off at the very beginning. So it took 2 to get it, and we produced 4, 2 for each one. So we have a net gain of 2 ATPs. Okay, so glycolysis produced 4 ATPs, but it took 2 to kickstart it. Remember in the beginning we talked about that. So it produced a net gain of 2 ATPs. This is outside the mitochondria. Okay, this is your mitochondria. This is where it enters into the mitochondria here. So this is glycolysis starting. Glycolysis broken down to 2 pyruvates, which produce 4 ATPs. It took 2 ATPs to kickstart the glycolysis, so we have a net gain of 2 ATPs. That's where this comes from. And it produced 2 NADHs. Each NADH produce 2 ATPs. It is outside the mitochondria. Okay, you don't have electron transport chain. It only produces 2 ATPs, and there's 2 of them. So it produces 4 ATPs. So far, so good? All right, got a thumbs up. Cool. All right, so you got 4 ATPs there. Now we enter the mitochondria. So we got 2 pyruvates, go to 2 acetyl coenzyme A's. And then 2 NADHs are produced. Okay, 1 for each of the acetyl coenzyme A's. And now we're in the mitochondria.
Crossing the threshold right there.
What's that?
Pyruvate, and that second step that you underlined underneath the line is it crossing the threshold to react?
No, this is crossing into the mitochondria now. That's
Okay.
just mitochondria. Okay, the pyruvate enters the mitochondria and belongs to the acetylcoenzyme A.
I'm sorry, you kind of cut out there. I didn't hear you.
Oh, the pyruvate crosses into the mitochondria and works with and joins with the acetylcoenzyme A.
Okay.
Now it's inside the mitochondria.
All right.
So it's going to be more energy efficient. So the 2 NADHs produced from the 2 pyruvate, 1 from each one, now produces 3 ATPs. So that gives you 6 ATPs. Okay, because each one produces 3 because it's inside the mitochondria now. Then it runs in the citric acid cycle, and it's going to make 2 turns in the citric acid cycle because there's 1 pyruvate and another pyruvate. So you got 2 of them. So instead of making 1 GTP, it produces 2 GTPs. And each one of those, each 1 GTP produces an ATP. So you got 2 of them, so you produce 2 ATPs. And as you run the citric acid cycle, you're producing 6 NADHs. Nicotine adenine dihydrogenase. You're producing 6 of those. And each one produces 3 ATPs. Okay, so you do 3 from each pyruvate. So you get a total of 6, and each one does 3. So 6 times 3 is 18 ATPs. And you've also produced 1 FADH2 for each one, so 2 of those. And FADH2, remember, doesn't do the first transport, but does the last 2. So it produces 2 ATPs for each one. So that gives you 4 ATP. So what you've got, okay, when you run 1 molecule of glucose from glycolysis through the citric acid cycle, you produce 36 ATPs. And that's where they come from.
Is there ever going to be a case where the citric acid cycle is only one term?
Say that again.
Is there ever going to be a case where the citric acid cycle is only one turn?
Yeah, I mean, it only goes one turn, but it goes one for each pyruvate produced. Now, say you're producing pyruvate and you shut down the citric acid before the second one runs through it. You only run one. So each one pyruvate is going to generate 18 ATPs.
Okay.
But you get 2 of them from every glucose molecules. So in terms of pyruvate, you're generating 18 ATPs. In terms of glucose, you're generating 2 pyruvates and you're generating 36 ATPs.
So is that a question that would be on the exam? Like if you started out with 4 pyruvates, then it would be 36 doubled?
Yeah, that's a very good question for the exam. Okay. But it'd be simple. It would say 1 pyruvate or 1 glucose. So 1 glucose is going to generate 36 ATP. 1 pyruvate is going to generate 18 ATPs. See, this is where you have to think about it. And you're not just memorizing numbers. Does that make sense?
Yeah, because
that's not going to word the question on the test.
Yeah. Okay.
I'll either give you 1 glucose and it's going to generate 36. I'll give you 1 pyruvate. It'll generate 18. And you can see how through that list right there. Okay. Any other questions on the citric acid cycle and this pyruvate going through it from glycolysis?
I have a question.
Sure.
Have we gone over exactly what ATP, how much ATP we need to do things? Like
everybody's different. You need ATP there's 2 things the body needs to contract a muscle. Remember, your heart's a muscle. There's 2 things you have to have my muscle contract. You have to have ATP and you have to have calcium. So it depends on your muscle activity. The more you're going to utilize, the more ATP you're going to need. The more you work out, the more active you are, the more ATP your body needs. Because to contract a muscle, no matter what that muscle is, you have to have ATP and calcium.
So for people that are like super athletes, for example, like sometimes we'll get patients that do like just have been professional athletes and stuff like that.
Right.
So their resting heart rate will be super low. Are they using less calcium? Or
no,
is calcium
they're still using calcium. You got to remember those athletes have quite an appetite too. You ever been around super athletes, like Olympic athletes? They eat. They have to provide calcium and nourishment for the cycle to keep going. They use a lot of ATP. They use it very efficiently. Okay, they're breaking down their body fat. Okay, they're using that to produce ATP because they used up a lot of it. It's not being stored. See, when ATP, when your body reaches a maximum, and your ATP you don't need any longer because you're not using it, then the proteins and the fatty acids that doesn't go through the ketone cycle. It goes into glucagon. It gets stored in your liver. It gets stored as body fat, as lipids, as triglycerides, and that stores your body fat. So if you're using like an athlete, ATP, you're generating a lot more ATP, but you're using it all the time. You're not storing that body fat. You're using up all your diet, all your food to generate this citric acid cycle. It keeps spinning. Okay, so depending how active you are, depends how much ATP your body needs.
So you're essentially producing more because you are more active.
Exactly. And you're not letting that food that you eat, those carbohydrates and those fats and proteins, you're not letting it go into fat storage. You're using it right away. You're not stopping your citric acid cycle. You're keeping it going because you are constantly producing ATP.
Okay, I was just curious about that because I know the calcium has a lot to do with like the efficiency of the heart.
Sure it does.
And we have, like I said, those patients, their resting heart rate will be like 40. And then they're just.
That's a marathon runner.
normal.
Yeah. If you got a resting heart rate of 40, you're running marathons. You're something extremely active. And you're excellent shape.
Yeah. I
Or
mean,
you got a disease. What do they do?
no, I mean, like I was talking in terms of like people that are healthy, because we do get those people that, like I said, they'll be like 60 and be like, oh, I'm preparing for my other triathlon, or they'll be like 70 and they're like, I'm gonna go run the whatever. There's some like marathon thing in Boston,
Yep.
New York.
The Boston
like,
Marathon. Yep.
Like send someone in the room because their heart rate's like 30 and they're just like asleep fine.
Yeah, you gotta watch it, because I mean, trained athletes can have a lower heart rate sometimes in the 40s, but usually in the 50s. But most other people in the 60s and 70s. But if it starts getting too low, you got problems also. Other things can cause that. Okay? Because you got to remember, to get calcium, if you're not eating right and your body needs that calcium, it's going to start breaking down your bones to get it. Because your bones are storage for calcium.
Right.
So osteoporosis, other things like that are for people that they're not getting enough calcium and their muscles need it. So it starts breaking down their own bones.
Okay.
Okay. So you have to have those two things required to contract a muscle, any muscle in the human body. You have to have ATP and you have to have calcium. That's what causes the muscles to contract. Write a balanced equation for the reduction of NAD. Remember, NAD goes to NADH. Okay. So you get NAD plus, goes from H minus, which is a proton with an extra hydrogen with 2 electrons now, goes to NADH. There's your balanced chemical equation for the reduction of NAD plus. Should be a plus sign right there. I can see if it's down below. Okay, so there's your balanced equation. Write a balanced equation for the reduction of FAD. FAD goes to FADH2, remember? Okay, so FAD plus 2 H pluses, plus 2 electrons. Okay, goes to FADH2. Okay, so those are the two reducing oxidizing agents that cause reduction. Okay, these ones that are being reduced, that are giving the electrons back into the electron transport chain to generate ATP. So FADH2 enters at the second and third transport spots. NADH enters at the first, second, and third transport spots. So FADH, for every FADH2, you get two ATPs. For every NADH, you get 3 ATPs being formed. Okay. Calculate the number of ATP produced by the complete oxidation of 1 molecule of glucose. So glucose is going to be how many pyruvates?
2.
2. How many spins of the citric acid cycle?
2.
2. So the spins were twice. Okay, and it's just what we calculated before. Glycolysis gives you a net of 2 ATPs, and then you got 2 NADHs, gives you 4 ATPs. So here's your 6 before you enter into the mitochondria. Into the mitochondria, you get 2 NADHs produced at 3 ATPs each. That's 6 because you remember you got 2 here because 2 pyruvates for 1 molecule of glucose. Okay. And each one gives you a GTP. There's 2 of those, gives you 2 ATPs, 6 NADHs because 3 from each one. Each one gives you 3, that's 18, and 2 FDs, FADH2s, because each pyruvate gives you 1. Okay. And that generates 2, so that's 4. And it's 36 is your total ATP. Okay. Represents about 40% of the potential energy of glucose. So breaking it down, about 36 ATPs from our glucose molecule. Okay. Now, calculate the number of ATPs produced by the complete oxidation of pyruvate. Okay. So now it's nothing, now it's pyruvate. So how many times are you going to spin the citric acid cycle with pyruvate?
1.
Once. Perfect. So the conversion of 2 NADHs, okay, give you 6 ATPs. Complete oxidation will produce 3 ATP molecules. Okay. So pyruvate goes to 2 acetyl coenzyme molecules. Okay. You're not running the citric acid cycle yet. but it says just complete oxidation of pyruvate. Okay. So you're going to put 2 NADHs for every 2 is going to produce 3. That gives you 6 ATPs. If you run from glucose like we had before, we have the 2 pyruvates. So if you only ran 1, you would have 3 ATPs, I'm sorry, as it enters it. Okay, this was right from right. Let's go back to right here. Okay, this is what we're talking about. This is a pyruvate molecule. Okay, so conversion of 2 pyruvate molecules to 2 acetocoenzyme A molecules, what they're asking. And we ran 2 of them, we produced 6. You run 1 of them, you're going to produce 3. So the question is asking how many ATPs is run by converting pyruvate to acetocoenzyme A. Okay, oxidation of pyruvate. And it's going to be 6 ATPs you're running 2, so 3 if you're only running 1. Okay. Sometimes we break down amino acids for energy. We don't like to do that, but if the body is starving, we can produce energy by breaking down our amino acids, by destroying our proteins in the body. Okay. When the body has depleted glycogen, which means we are out of glucose storage, by starving, we are going to start using amino acids for fuel. Okay, degradation of amino acids take place in the liver in 2 stages. Right? You remove the alpha amino group, okay, and you break down the carbon skeleton into a variety of different compounds. Go to pyruvate or acetocoenzyme A, which will help you produce ATP. Okay. The amino group that you take off the amino acid is excreted in the urine, and it goes through the urine cycle, which we'll talk about that too. Okay. So removal of the alpha amino group is transamination. We were transferring the amino group from one thing, the amino acid, to another compound now. So transaminase is the enzyme that catalyzes the removal of or the transfer of your alpha amino group to another compound. So transaminases catalyze the transfer of alpha amino group from alpha amino acid to an alpha keto acid. Remember, keto acid has a ketone function, which is a carbonyl group. The alpha keto acid is often alpha keto glutarate. Transfer process appears simple, but it's actually really complex. Transaminase binds amino acid to its active site, transfer amino group to what's called pyridoxal phosphate. Then from the pyridoxal phosphate, it moves amino group to a keto acid. So the pyridoxal phosphate is an intermediate step. So this is your amino acid. Okay, this is the keto, accepted keto acid. It's going to accept this nitrogen group. Okay, this is the alpha keto acid of amino acid. Okay, and then this is a new amino acid that's formed. Okay, so you transferred it from one to another. Okay, and then you can break that down. The pyridoxal phosphate was our intermediate, and pyridoxine. Pyridoxal phosphate is a coenzyme required for all transamination reactions. So all the amino acids are being broken down. We use pyridoxal phosphate as the intermediate step. Okay, there's over 50 discovered. All use this coenzyme that break this process down. Pyridoxine is vitamin B6, okay, which is the source of pyridoxal phosphate. That's how we get it, by taking in vitamin B6. Okay. Aspartate transaminase. This particular transaminase catalyzes the transfer of alpha amino acid of aspartate to alpha ketoglutarate. And it produces oxaloacetate and glutamate. Oxaloacetate is our starting point for the citric acid cycle to produce ATP. So we take aspartate as amino acid, we react it with our alpha ketoglutarate. Okay. We produce oxaloacetate and glutamate. So we transfer, it's a transaminase. We transfer the ammonia group, NH3 group, our alpha amino group, to another substance.
Is aspartate equivalent to the sugar substitute that they use?
That's what it's same name, yeah.
Okay.
This is the amino acid aspartate. But that's what makes up aspartate as your sugar substitute. That for aspartate. Now alanine. Alanine is another amino acid. Here's alanine. Okay. Catalyze the transfer of the alpha amino group of alanine to alpha ketoglutarate again. Okay, it produces pyruvate and glutamate. Again, so here's your alpha ketoglutarate. Okay, and it's going to give you pyruvate and glutamate. So you transfer it and you're creating components to run through the citric acid cycle to produce ATP. So you produce pyruvate and glutamate. The transaminase produces glutamate, are called transglutamases. Produces citric acid cycle intermediate. So it produces parts of the citric acid cycle that are going to help run to produce more ATP during times of starvation. Okay. Provides a direct link between amino acid degradation and the citric acid cycle. So it allows you to break down your amino acids. And like one of the signs of starving is you break down your proteins. Your body becomes very thin. You start seeing your skeleton because your proteins are being broken down, because your amino acids are being broken down to produce energy, because you're not taking enough food to produce energy. So what this does provides a direct link between your amino acid degradation and the citric acid cycle, because we're oxaloacetate, pyruvate, we're generating intermediates inside the citric acid so we can get that kick into the citric acid cycle and produce ATP. So your amino acids are being broken down and you're forming ATP for energy. When your amino acids break down, you break down your proteins and your body becoming out of whack, basically. Okay. So what is the role of pyridoxal phosphate? Okay, pyridoxal phosphate is our intermediate. Okay. During transamination reactions, the amino acid group is transferred to the coenzyme pyridoxal phosphate. That is our intermediate in all transamination reactions where we're losing our alpha amino group from our amino acids. In the last part of the reaction, then the amino group is transferred from the pyridoxal phosphate back to an alpha keto acid. Okay. Transaminase allows you to transfer your nitrogen group, your alpha amino acid. So transaminase is an enzyme, the transfer of an amino acid group from an amino acid to a keto acid. Okay, so oxidative deamination is removal of your alpha amino group. So ammonium iodine, ammonium ion is liberated from the glutamate form from the transaminase reaction. Glutamate breakdown is catalyzed by glutamate dehydrogenase. So you start with glutamate, which is your amino acid, or one of the amino acids. Okay, you're going to form free ammonia. Okay, and this is your alpha ketoglutarate again, and you're forming NADH, which is going to go along and form into ATP, producing energy. Now, so when you're breaking down amino acids to form energy, you're getting free ammonia into the system. Ammonia is extremely toxic to the human body. You got to get rid of it. You got to get rid of it quick. Okay, and that's what we do. Oxidative deamination is an oxidation reduction process where NAD plus reduces NADH, and this gives you ATP. The amino acid is deaminated, so the amino group is removed, and it produces ammonia ions in the body, NH4, which is not a good thing. Okay, but this is how pyridoxal phosphate works. This is a pyridoxal phosphate enzyme. It starts with the bond like aspartate is your amino acid. Okay, comes back, it produces oxaloacetate because it transfers this amino group to here. Now you got the amino group here, and then the alpha ketoglutarate comes in. You transfer the amino group back to that to form glutamate, and then the pyroxidal phosphate is regenerated, and the next one can come back in. So you generate your enzyme, your catalyst over and over, and it's just transferring the amino group. That's what pyridoxal phosphate does. Transfer the amino group to an alpha ketoglutarate. Okay. So your alpha amino, this is your alpha amino acid, and it goes into alpha keto acid. You into an alpha ketoglutarate, which it bonded to form glutamate. Okay. Oxidative, you've taken NAD to NADH for energy, and you've produced NH4 plus. NH4 plus is very toxic to the human body, like we said, and it has to form urea. Urea is produces urine, and it's removed from the body. You can be very careful with that NH4 buildup. Okay, so this is the fate of different skeletons. These are all your different amino acids: leucine, tyrosine, lysine, glycine, and you can see where they enter into the citric acid cycle at different stages. Like I said, histine, proline, all the amino acids can be broken down through pyridoxal phosphatase, okay, and enter into the citric acid to produce energy. And they all enter, well, not all, but some enter at different sites, some enter at the same site. But they're all going to produce an intermediate that is accessible to entering the citric acid cycle to produce ATP. The urea cycle, oxidative deaminate, because we produce NH4 plus, right, which is toxic. So we've got to get rid of that. We got to run through the urea cycle now to get rid of NH4 into the form of urea. So oxidative deamination, we took the ammonia group off of our amino acids, produce large amounts of ammonium ion. Ammonium ion is toxic, okay? Must be removed quickly from the body. It cannot build up. No matter how much energy your body needs, if it allows ammonia to build up, your body's going to cease to function. It's extremely toxic in your body. So eliminated ammonium ion is used through the urea cycle. Urea is then excreted in your urine. Failure of enzymes in urea cycle can be genetic and lead to what's called hyperammonemia. Hyperammonemia. Okay. In other words, the buildup of ammonia in the body. And it leads to early death. Okay. Toxic ammonium ion buildup leads to retardation, convulsions, and vomiting. This is an overview of the urea cycle. These are the ones involved, and we'll go through this step by step. Okay, this is the overview of it. And the first step is going to be combined carbon dioxide and ammonia. So ammonia is produced, okay, it's combined with CO2, okay, to form carbonyl phosphate. This is the result of CO2 and ammonia. And it takes 2 APTs to do this. So it takes energy to do this. But even though if your body's still requiring energy, you have to do this, or NH4, the buildup of ammonia is going to kill you. So it requires ATP and water. It takes place in the mitochondria. It is catalyzed by carbonyl phosphate synthetase is the enzyme. And again, your carbonyl phosphate is a product. And you went from ATP to ADP, free phosphate, and then 3 hydrogen. Okay. Then it takes a carbonyl phosphate, okay, condensed it with amino acid ornithine to produce amino acid citrulline. Okay. Again, it occurs in the mitochondria. It's catalyzed by ornithine transcarbamylase. That's your second step. Okay, because here's your carbonyl phosphate. This was generating the first step. And this came from your first step. And then it's combined with ornithine to produce citrulline. Okay. The next one, citrulline, okay, was produced in the step above. It's transported to the cytoplasm. So it goes out of the mitochondria now, condensed with aspartate to produce arganosuccinate. That's your product here, arganosuccinate. Okay. And it's catalyzed by arganosuccinate synthase. Okay. It requires energy released by ATP hydrolysis. So again, you're breaking down ATP for this reaction to occur. So you're using energy to run the urea cycle. You're not making energy. Okay. But you have to run it because you have to get rid of the nitrogen. And again, you're taking this nitrogen compound and you're getting it more and more complex. So you can get rid of that nitrogen. That's what your main goal is. Then you take the arganosuccinate, okay? It's cleaved, broken into 2 pieces by the amino acid arginine and fumarate. Okay? Fumarate from the citric acid cycle, and it's catalyzed by arganosuccinate lyase. So here's your compound, you're breaking this off right there, and you're forming arginine, your amino acid, and fumarate. Okay, then arginine is what you formed, adding water to it to form urea. Okay, and you produce ornithine, regenerating the ornithine again to use back again at the start of the reaction. Okay, so this is what you generate as urea, and this can be excreted by the body in the urine. Okay, so what's the purpose of the urea cycle? To get rid of ammonia. Basically, the purpose of the urea cycle is to convert toxic ammonia ions to urea, which gets excreted in the urine. Okay, where did the reaction in the urea cycle occur? Okay, it occurs in the liver. Overview of anabolism. Remember anabolism and catabolism. Catabolism is breaking up. Anabolism is reforming it, biosynthesis. Okay, citric acid cycle is a source of biosynthetic intermediates. We can make compounds from the citric acid cycle. So the citric acid cycle functions as an energy harvesting mechanism. We could produce ATP, we harness energy, we make energy. Anabolism is creating, biosynthesis, making compounds. We use some of our intermediates in a cycle to make certain compounds that we need. So as amino acids can be converted to citric acid cycle, intermediates that we just saw, they can be used to make, these can be used to make amino acids. So we can use our citric acid cycle to make amino acids also. So we can use it to break down amino acids, come into as intermediates. We can also use the intermediates to, once body establishes what it needs, it can go back to making amino acids. Oxaloacetate is used to make aspartate. Oxaloacetate is our starting point for the citric acid cycle. We can use that to make aspartate if we need it. Asparagine is made from aspartate in an animation reaction. Okay, we aminate, we move the amino group to aspartate. Okay, glutamate can be made from the alpha ketoglutarate that was used. So glutamine, proline, arginine are all made from glutamate. Okay. So again, you can see using the citric acid cycle. So instead of oxaloacetate going back in the cycle, we can use it to make aspartate, which makes the pyrimidines, which is RNA, DNA, our bases, and aspartate. Succinylcholine. Okay, instead of going into succinate, it can be used to make porphyrin rings for our heme systems, for the red blood cells. Okay, so we use the different intermediates. You can see where the 3-phosphoglycerate can be made into it. Glucose 6-phosphate can go to ribose, can go to tyrosine. So we can use basically what we're saying is running the citric acid cycle, glycolysis citric acid cycle produces ATP. When we have abundance ATP, we can use that cycle to produce some of the amino acids that we need. Okay? So we don't have to, we can run it and not produce all the ATP that we can. We can push off to the side some of these intermediates and make the amino acids that our body needs. Okay? Now that being said, there are some amino acids we cannot make. Those are the essential amino acids. And those you have to get from your diet. You cannot produce those. But these are the ones we can produce by the different side reactions that can occur and just not run the citric acid cycle. Remember intermediates from it. So explaining how the citric acid cycle serves in an amphibolic pathway, just like we just said. The amphibolic pathway is a metabolic pathway that functions both in anabolism and catabolism. We break it down to produce ATP, or we can use those intermediate steps to produce more amino acid and biosynthesize them. So the citric acid cycle is amphibolic, can do both, anabolic and catabolic, because it has catabolic functions, completely oxidize acetyl acid to provide electrons for ATP synthesis. So it breaks down everything, down all the way oxidation, into CO2 and water to produce ATP. Or we can use the immediates as precursors for biosynthesis of other molecules. Okay, that's anabolism. So we can use the citric acid cycle can function as both. And you can see how it can do that with the intermediates that are produced. What is the function of anapleutaric reactions? Okay, anapleutaric is one that replenishes an essential intermediate like oxaloacetate. Okay, it's in the reaction that replenishes what you need to start the citric acid cycle. So a critical biochemical pathway like the citric acid cycle can continue to function. So it replaces things you need to get that citric acid cycle started to break those intermediate products. Okay, so you have to have these anapleutaric reactions to make sure you don't run out of oxaloacetate or other components that are necessary for the reaction to occur.